All right, hello everybody. This is going to be a presentation on three-point test crosses. It was done by Arjun, Ashley, Clay, Krisha, and myself, Marco. So, what is a three-point test cross? The three-point test cross is a genetic technique where an organism which has been identified as being heterozygous for three genes of interest is crossed with an organism which has been identified as being homozygous recessive for those same three genes. Three-point test crosses are more accurate than two-point test crosses, especially at determining long distances. Three-point crosses also account for the effects of double crossover events. What can three-point test crosses do? So, by analyzing recombination frequencies between each pair of genes, geneticists can determine the distance between genes as well as their order on a chromosome. So with that information, we can develop a gene map. When do we use three-point test crosses? So they're very useful when mapping a new mutation whose location is unknown relative to two already known mapped genes. By analyzing the progeny phenotypes and comparing with parental gametes, we can differentiate the recombinant and parental gametes and calculate recombination frequencies. Doing so allows for the identification of double crossover events and their frequency. Generally, to work through a problem, I like to break it down into 4 steps: identifying the parental and double crossover gametes, identifying the gene order using recombination frequencies to determine gene distances, and then constructing the gene map. All right, so we have our question introduction here. A true breeding tall plant, which produces green wrinkled peas, is crossed with a true breeding dwarf plant, which produces yellow round peas. And they produce a heterozygous yellow tall round pea plant. That's our F1 generation. And this F1 plant is now crossed with a homozygous recessive pea plant with regards to plant height, pea color, and shape. The three-point test cross produces offspring with the following phenotypes, and we can see them down below. Right, so identifying parental and double crossover gametes. Normally you won't be told which are the parental gametes and which are the double crossovers, so you'll have to be able to figure that out just based on their frequency that you see them in the data set. So the ones that you see the most are going to be parental here. The ones you see the least are going to be double crossover. And then the rest will be single crossover. All right, so now we're going to identify the gene order. So we're going to compare the double crossover gametes to the parental gametes here and here. And we can then match the loci to determine which one is the odd one out. So in our case, we see that big T and small r are the same in the parental top two, and small t large r and small t large r are a pair found in the bottom two here. So that means between parental and double crossover, Y is the gene that is being switched, and as a result, that's the one that we know is in the middle. So now we're going to determine the distance between genes by calculating their recombination frequencies. So to determine the distance between the genes, Y to T, Y to R, and T to R, we calculate the recombination frequencies by adding the sum of the crossover gametes with respect to just two of the genes of interest at a time. And then we divide by the total gametes and multiply by 100%. So when we're looking at the distance from Y to T, we want to look at the parental gametes and just look at Y and T and sort of just disregard R for now. And we see that it appears as small y and big T and big Y and little t. So all of our crossover with respect to those two genes, Y and T, are going to have a different setup there. So we're going to look for either little y, little t, or big Y, big T. And then we're going to add those all up and divide the sum of all of the gametes. So in this case, that's the double crossovers, 8 plus 4. And then we also see big Y, big T, and little y, little t. So 92 and 76, 92 and 76. And then the total for this data set is 2000. So divide by 2000 times by 100%, and we get 9% or 9 map units. If we look at y to r, we're going to look here. We got a small y, small r, and a big Y and a big R. So all of our crossovers are going to have either small y, big R, or big Y, small r. So we see that here in the double crossovers, 8 and 4, 8 and 4, and in these single crossovers here as well. So big Y, little y, big R. So 126, 142, 126, 142 divided by 2000 times 100%, and we get 14% or 14 map units. And then for T to R, same kind of process, and we get 21.8 or 21.8 map units. All right, so now we're going to assemble a gene map. So first you need to know the gene order. We've already determined that. And second, you need to know the recombination frequencies, and we've already calculated that as well. So now you just need to set up the order of your genes. We have ours R, Y, T with R or Y in the middle, sorry. We could also have it set up as T, Y, R. It wouldn't matter either way, just as long as Y is in the middle. And then we use our recombination frequencies to represent the distance in map units. So 1% frequency equals 1 map unit. It's also equivalent to 1 centimorgan. So you can see here we've got R, 14% or 14 map units to Y, and Y to T is 9% or 9 map units. Earlier, we also calculated T to R as being 21.8%, but it's important to remember that when we're determining longer distances, it's more accurate to add up multiple shorter distances than take one calculation for one longer distance. So in this case, we've gone with 14 plus 9 equals 23% or 23 map units instead of the 21.8. All right, so these are our references, and that is it for 3-point test crosses. Thank you very much.